Everybody, as you can see, they gave us a new clock, one that's actually able to keep time, it looks like, so it's wonderful. No major announcements. The weekly quiz in here, or not the lecture quiz in here on Friday will be on photosynthesis, which we are going to finish up today. And we'll probably start the next chapter, which is cellular respiration, but that will not be on the Friday quiz. Well, all right, let's see. So this is, I guess, a good place to start. So this is showing the chloroplast organelle, which is the organelle inside mesophyll cells where the photosynthesis takes place, and it's illustrating the two parts of photosynthesis, the light-dependent reaction, which takes place in the thylakoid membrane, which the folds of which are sometimes called the grana. So sometimes people would say the light-dependent reaction takes place in the grana. Come on, laser pointer. There we go. Some people say, well, it's taking place in the grana, but you could say thylakoid membrane. Either one is fine. And the point of that is that the energy in the sunlight is changed into some chemical potential energy in ATP molecules and NADPH molecules. And those drift away from the thylakoid membrane into the stroma, which is the watery liquid that fills the chloroplast. And then eventually they meet the enzymes of the Calvin cycle. And a cycle is a circular metabolic pathway, a group of enzymes that carry out changes on molecules in a circular fashion, so to speak. And what the Calvin cycle is doing is it's fixing the carbon dioxide that comes into the leaf. And fixing means not that it's broken, but that the carbon dioxide is going to become joined to other molecules and get changed into glucose molecules, which is the whole point of the photosynthesis. Is there a question? Oh, the lights? Yeah, okay. All right. I was getting there. All right, so let's dissect the Calvin cycle in a little more detail. Here's a figure from your textbook that shows the Calvin cycle. You can see the carbon dioxide coming in here at the top, and the glucose is being made down here at the bottom. Arrows represent enzymes in cycles and metabolic pathways. They only show one arrow here, but there's actually several enzymes that are part of the Calvin cycle. And the molecules that you find within a metabolic pathway are called intermediates of that metabolic pathway. And so we're going to be focusing in on a few of the intermediates here. There's an intermediate called ribulose biphosphate, or RUBP for short. It's a 5-carbon molecule. It's actually a type of sugar. And to show the carbon atoms in the molecule's backbone, they're just showing these little gray circles. And of course, it has other atoms. RUBP has hydrogens and oxygens that are attached to those carbons, but you can get a pretty good understanding of the Calvin cycle if you just focus on the carbons of the molecules. So that's what we'll be doing. So C5 means a 5-carbon molecule. RUBP is a C5 molecule. Another molecule we're going to look at is something called phosphoglyceric acid, PGA. It's a C3 molecule. Another molecule we'll look at is called PGAL, phosphoglyceraldehyde. It's also a C3 molecule. And of course, each carbon dioxide molecule is a C1 molecule. All right, so those are the intermediates that we'll be looking at. This is how I'm going to represent the Calvin cycle. Again, the green arrows are going to represent enzymes. Now I'm going to start off with showing 3 of those RUBP molecules at this point here in the Calvin cycle. We'll consider this the beginning of a Calvin cycle, but in a way that's kind of a strange thing to say because circles really don't have a beginning or an end. But just to begin our discussion, we'll start at this point in the Calvin cycle. Now, in real life, inside the stroma of a chloroplast, I'm sure there are more than 3 RUBP molecules floating around. There's probably thousands, if not millions. But just to discuss the Calvin cycle, let's just show 3 of those going through the cycle. So notice that at that part of the cycle, the grand total is 15 carbon atoms. Each RUBP has 5, and we're showing 3, that's 15 total. Now that first enzyme of the Calvin cycle, this one right here, what it's going to do is to grab one of those RUBPs and attach the carbon from a carbon dioxide to it. So to illustrate that enzyme like this, it has a binding site for carbon dioxides, and it also has a binding site for RUBP molecules. Maybe I'll use different colors here. So this black blob is that first enzyme. It can grab a carbon dioxide, it can grab an RUBP, it has binding sites for both. Matter of fact, I think I've got that here. So it grabs an RUBP, it grabs a carbon dioxide, like that. And then what it does is carry out a chemical reaction where it clicks them together. So what comes out is actually a C6 molecule. Made out of 5 carbons from the RUBP and the 1 carbon from the carbon dioxide. But as it turns out, that C6 molecule that's first made by this enzyme is very unstable, meaning it breaks apart very easily. And so almost instantly, it breaks apart into 2 C3 molecules, which are phosphoglyceric acid. And so, think about it, the accounting of the carbons makes sense. We had 5 carbons from the RUBP, 1 carbon from there. They were brought together, that gives us 6. The 6 splits in 2, so you get 3 and 3, which is still the 6 that came into this particular enzyme. Now, so you don't have to know the name of this enzyme that's fixing the carbon dioxides, but I'm going to refer to it a couple times. So I'll give you the name for it now. Its official name is Rubisco, which sounds like it'd be a good name for a dog or something. Rubisco, like that. But it comes from ribulose biphosphate carboxylase is where they got its name from. Anyway, so that's the actual, Rubisco is the actual enzyme that fixes carbon dioxide within the Calvin cycle. That green arrow right there is Rubisco. All right, let's watch it do its thing. So it brings together the ribulose biphosphate and this carbon from the CO2, briefly forms a C6 molecule, but that breaks apart instantly. So you really end up with 2 molecules of PGA. And so if I ask you, what is the first molecule that carbon dioxide is fixed into, you should answer PGA, because this one doesn't count, it's not a stable molecule. Okay, so we used up one of our RUBPs and we ended up with 2 PGA molecules. The next step is this enzyme right here that's going to take those PGAs and modify them slightly. Here's a phosphoglyceric acid molecule. Don't worry about memorizing its structure, just introducing you to it. All right, so that enzyme there grabs a PGA, and using the energy of ATP and NADPH, it changes one of the functional groups, and you end up with a molecule called PGAL, phosphoglyceraldehyde. Here's what PGAL looks like. Now, if you have really good recall, you'd be able to say, aha, I see some functional groups have been changed. And here they are. And if you remember your oxidations and reduction, this is a reduction reaction. because this carbon here at the end is losing a low energy carbon-oxygen bond and replacing it with a higher energy carbon-hydrogen bond. And when you're talking about carbon atoms, all reductions are non-spontaneous, endergonic uphill energy reactions. And so that is why the Calvin cycle needs ATP and NADPH. That enzyme needs energy to push this reaction uphill, so to speak, to make it happen. All right, so one of our two PGAs has become a P-gal. Which is still a C3 molecule. And now the other PGA is going to go through that same enzyme. So again, it's going to need ATP and NADPH to power it. And we've got another P-gal. All right, so notice that, oh, now it's going, stop. Computer. Oh, looks like it wants to go automatically. Well, anyway. It won't let me stop. Well, oh well. So anyway, the point I was going to make is that notice for every one RUBP that went through this part of the cycle, we ended up with two P-gals. And so now we'll watch the next RUBP go through. Same thing, that Rubisco enzyme fixes carbon dioxide onto it, so you end up with two PGAs. They go through that particular enzyme and get changed into P-gals. So we end up with two more P-gals. And then lastly, this RUBP is going to go through that part of the cycle. There we go. Carbon dioxide is fixed to it with the Rubisco enzyme. Two more PGAs, and they get reduced. Both changed into P-gal. All right. So we started off with three RUBPs here, and we ended up with six P-gals there because each RUBP ends up generating two P-gals. So we had three there, and 3 times 2 gives us the 6. Okay, now to keep a cycle going, you need to regenerate the intermediates that you used up. So we need to regenerate those three RUBPs. How many of these P-gals do we need to do that? Not 6. We have to regenerate those three RUBPs that we started. How many of those P-gals do we need to regenerate the three RUBPs? 5 of them, because the three RUBPs were how many carbons? Not 5. The three RUBPs were 15, right? Each RUBP is a C5 molecule. And since remember we started with 3 of those RUBPs there, that's 15 carbons. So we need to regenerate 15 carbons. And so how many of the P-gals is that? It's 5 of them. Here we go. So out of every 6 P-gals that are generated, you need to get 5 to regenerate the starting RUBPs that you had there. And that's what these enzymes are going to do right there. Those grab P-gals and essentially break them into smaller bits and reassemble the RUBPs out of them. And that's good. By reassembling our starting materials, that allows the cycle to keep turning. And so at first you may say, well, what's the point? We started with 3 RUBPs and we go around, we end up with 3 RUBPs. Well, what good does this do us? Well, notice that we have an extra P-gal that didn't need to be regenerated. And so that is a new organic molecule that didn't exist inside the plant before. 5 out of the 6 P-gals do have to be used to regenerate RUBPs, but the 1 out of the 6 P-gal is extra and doesn't have to be used to regenerate RUBPs. Now, there's sort of a mystery here. You've probably heard this law of science, conservation of mass. where did these 3 atoms come from? You know, how did we start with 15 and suddenly end up with 15 plus 3? Who can answer that for me? From the carbon dioxides, right, because remember this Calvin cycle is not a closed system. Every time an RUBP goes through this part of the cycle, a carbon dioxide comes into the cycle. So when these 3 RUBPs went through this part of the cycle, 3 carbon dioxides came into the cycle. And so in essence, that's how these 3 extra carbons came from. That's where they came from. In essence, the Calvin cycle has changed the 3 carbon dioxides that came in into 1 new P gap